Hi, everyone. I'm Xuegong Zhang from Tsinghua University. It's a pleasure to share our recent work on human ensemble cell atlas as a virtual body for in-data cellular experiments. So we are all composed of cells with virtually the same set of genes. The differential expression of genes in our cells makes us a living human body rather than just a lump of cell clones. Therefore, building the molecular atlas of human cells using the current and future high-throughput single-cell technologies is a key for better understanding of life in health or disease. So the motivation of this work is a question what the cell atlas should be alike. We started to ask about 5 years ago. Looking into the data portals of the major cell atlas projects like the Human Cell Atlas or hECA, we found that the question is still not answered yet now. So most of the current practice are actually huge archives of single-cell data files indexed by information of the studies that generated the data. So referring to the Human Genome Project, data were first deposited and archived at the Gene Bank and other data portals. So they are similarly, they are huge archives of data files. But the genome was finally assembled and presented at the UCSC Genome Browser and other sites as a unified single repository indexed by the genomic coordinates. So currently, the cell atlas has not yet been assembled. So remember the strategy used for finally assembling the human genome, the shotgun assembly. So we ask the question, can we assemble cell atlases from scattered data in a similar shotgun manner? So for the assembly, we need a unified framework to organize all the information. The human genome has the 1D coordinate as a natural backbone of the information framework. What is the natural backbone of the organization of information of cells of our body? We may think that it would be the 3D anatomic structure of the human body at the beginning. However, with our efforts of constructing the adult human heart cell atlas of multiple anatomic parts, we realize that there are multi-facet, multi-resolution, spatial and functional heterogeneity among our cells. So the heterogeneity exists in spatial, temporal, and functional dimensions at multiple granularities. A single set of 3D or 4D or even high-dimensional coordinate system is not sufficient to represent the heterogeneity. On the other hand, the spatial location of specific cells are not destined to be indexed by or fixed by a coordinate, a specific coordinate in the system. So with these observations, we propose a unified information framework for cell atlas assembly. That is, all cells should be stored in a big data repository that enables quick indexing of cells at all angles. And there should be a unified ontology graph for a unified annotation of cells. In other words, the cell atlas should be assembled as a virtual human body of all digital cells from our organs and tissues. To achieve this goal, we realize that the size of information that are needed to manage for cell atlas is multiple magnitudes higher than that of the genome. It turned out to be a huge challenge for the information technology. The current practice is to store and manage the files that we call it the file level collection. What we need to do is seamless cell level or cell-centric assembly to make the atlas as a virtual human body. So hECA, or the Human Ensemble Cell Atlas, is the solution we proposed and developed for cell-centric atlas assembly. Before going into the details, let me explain the cover figure we designed. So we designed the cover figure using the movable types in the language, in the printing as a metaphor. So the cells are like the movable types in a language. So a specific way of their usage and arrangement may compose a human organ or a human body. But there is not only one designated way of doing this. There are maybe also many other ways to organize the cell types. And there are many other ways to read the article of the human life from different angles at different levels when we are looking at the specific developmental or disease procedures. So we want the hECA to be able to provide a powerful, flexible way of assembling all collected cells into one entity, the virtual human body, like we assemble the movable types into an article or into a book. So at the bottom of the hECA is a database technology developed by my colleague, Dr. Rui Jiang. So we called the database as UGT, a unified giant table to store all cells from all sources or for all human organs. Its current capacity is more than 100 million cells, each of them with more than 100 million features that is on a single desktop computer. And all these cells and all the information can be retrieved efficiently within seconds. The second feature or technology of hECA is UHAF, a unified hierarchical annotation framework we developed as a standard knowledge tree or knowledge graph for the anatomic entities and cell types of the atlas. It is complete with regard to the data we have collected and expandable for future data. This right panel gives the website address and illustrates its homepage. So we designed this system as a platform for the community to contribute together to make this knowledge graph of cell types more and more complete. We welcome everyone to work with us together on this effort. The third technology or feature is ECCB, a programming interface that enables searching cells in the database either with a simple or complicated rule on gene expressions, on the metadata, on the annotation or other information available in the system. So in this way, cell sorting becomes a task of writing a few lines of computer codes. Therefore, we call it in-data cell sorting. I will come back to its applications in a few slides later. So with the UGT, UHAF, and ECCB, we built the hECA with more than 1 million cells collected from more than 100 scattered data sets. So these 1 million cells are from 38 healthy human organs. It can be seen as a toy version of the virtual human body we proposed. And we can experiment with many possible applications of this virtual human body, this assembled cell atlas. So the current version of hECA provides 3 basic functions. So for the interest of time, I will focus on the in-data cell sorting in this talk. So this is a website of hECA. We welcome users to all the audience to try it. So we know that in cellular biology, cell sorting is a basic step for many experiments. It is to select cells with specific properties, either from in vivo or in vitro samples for further study. So with the hECA we built and the ECCB feature we provided, so we turned this procedure of cell sorting to the writing a few lines of codes. And in these codes, we can include virtually any condition you want to define for your study. So virtually you can select any combinations or subgroups of cells according to their gene expression or their organ, the source organ, the annotated cell type, or even the technique used to get the data, or some properties or features of the donor, and so on and so forth. So this in data cell sorting is a term we coined with this paper. So the in data cell sorting is much more powerful than the conventional in vivo or in vitro cell sorting. So a straightforward application of this in data cell sorting is the agile construction of customized sub-atlases for particular scientific questions. So the cells of the sub-atlas formed by the in data cell sorting can be downloaded by the users, and users can apply whatever downstream analysis they want on this data for their investigation. No matter the data were originally from which study and from which technology. So a more advanced application of this in data cell sorting is in data drug experiment. So for many drugs, it is not okay to directly apply on the human body at the current phase. So we can apply it virtually on the ECA as a virtual human body if we know the effect of this drug on the gene expression. And with ECOT, we can collect the cells that would respond to the drug if the drug are really applied on the human body. We can study the effect of this drug on the whole human body by analyzing the collected cells with virtual responses to the drug. So this is a real example we did for the CAR-T drug for the CD19 positive cells and CD22 positive cells. The above panel here shows the result on the CD19 plus CAR-T drug. So from this panel, we know that all these cells have high expression of CD19, which indicates that they would be the targets of this drug if the drug is applied. And this marker indicates that only these cells in the red circle are the real targets of the designed target of the drug. All the other cells, about half of the total selected cells, they are off targets. And from the rich information in hECA, we can figure out what organs or tissues these off-target cells are. And by checking the marker genes that hECA provided, we can figure out what cell types or subtypes are these off-target cells. So in this way, we can predict the side effects of the target therapy. And all these analysis results are highly consistent with what has been reported in the literature. And also this experiment highlighted some possible new findings. So of course you may wonder that the idea is it's great, but the current collection of data is too small to really representing all cells of the human body. So of course we and the whole community actually are collecting more and more data. So we are expecting that with more and more data, this virtual human body will be more and more like the real human body. On the other hand, we also working on developing deep learning methods to kind of a complement to the data collected. For example, we can learn from the current data to generate more data. This is one of the work we are doing towards this purpose. So this model, we call it the Unicode model for data augmentation and data representation. It is a specially designed hybrid VAE or variational autoencoder deep neural network model. For the interest of time, I won't explain the details, but we have put the preprint on by archive. You can figure it out. So the model can learn an interpretable unified coordinated system for all the properties of the cells of interest. And with this model pre-trained on the collected large data, we can make the model to generate unobserved data along some of the properties we are interested in. So I will quickly show some example here. I won't be able to get into details, but you can figure out what the capacity of this model is. This example is an experiment to kind of convert the data measured from one technology to another technology. This is in this particular experiment, we regenerate the data from all different platforms so that the generated data are as if they were collected from the 10x platform. And this is an example that we generated T cells data from the data of actually other cell types. In this way, transfer the data to the T cells. And this is an experiment we did for generating cells in multiple organs along the vasculature coordinate. So that is a coordinate we discovered in our heart cell atlas work. And using the Unicode, we can generate more data, and we can see that we can control the position of the generated data along the coordinate. So this is an example we did for generating more data along a time trajectory. We know there is a limitation on the sampling time if we want to study a development procedure. However, we can use Unicode to learn the time trajectory and then to generate or interpolate many more new cells along the time trajectory. And this is a similar experiment, but applied on generating more cells along the local spatial coordinate. This is a local coordinate we found in our heart atlas study. So in this way, we can see that we can use neural network models like Unicode to kind of compensate on the shortage of the observed data to some extent. But of course, what we know about the cells transcriptomics, about the biology behind the cells transcriptomics is still limited. So there are many other things that models like Unicode may still not be able to learn. So we did some another work recently. This is a very the latest work. We collaborated with Biomap in Beijing. We developed a large-scale foundation model for single-cell transcriptomics. We called the model as SC Foundation. It borrowed the idea from large language models like GPTs in natural language processing. We use SC Foundation to learn the language, the transcriptomic language of the cells. And this model contains more than 100 million parameters, and it takes 20,000 genes as its basic token. The model is trained on more than 50 million single-cell RNA-seq data. So to our knowledge, it is currently the largest model of this type on single-cell transcriptomic data. This is also the largest training data set. Experiments have shown that this large model has many advantages in many downstream, in a variety of downstream tasks such as cell clustering, drug response prediction, perturbation prediction, and many other more tasks. For the interest of time, I won't explain the details. You can check out the preprint if you are interested. So summary. So we developed hECA, the Human Ensemble Cell Atlas. It is the first cell-centric assembly of cell atlas, and we developed the method of in-data experiment in hECA. This in-data experiment actually makes the cell atlas customized as cellular digital twins in the future for in-data clinical trials. We developed Unicode for unified representation of all cells and for data augmentation. We also developed SC Foundation, the largest cell model that shows a promising future for using current large models in AI to help to learn the underlying biology from the biological data. So actually, all these three work are examples at different levels of a new concept we proposed recently. The concept, we call it digital life or D-life. By D-life, we mean that to represent and reproduce life phenomena and rules measured by data, represented by datas and quantities, and with mathematical or AI models to describe the underlying biology and to reproduce the underlying biology. So with this, I want to conclude this talk. And most of the contents of this talk have been covered in these 4 papers or preprints. And this is the team and the students behind the work. I want to thank the funding agencies for their support and thank everyone for your attention. Thanks.